Okay, so now I'm honored to give the floor to Dr. Abhishek Kumar from Institute of Bioinformatics from India. He will give a speech on translational bioinformatics today. So, Dr. Kumar, if you're here.
Yeah, I am here.
Hi, welcome.
Welcome.
Welcome.
Hello, good
Thank
evening.
Hello.
We don't know about the time difference, to be honest. We say good evening, but
Yeah, it's evening only.
right, okay.
Yeah.
Professor, you can share your screen using the green
Yeah,
button
sure.
at the bottom of the screen. Okay.
You have to allow me. Okay.
Yeah,
share
right
screen
now.
option. You have to set the multiple.
Okay, we are giving now. Okay.
Yeah, now it's possible. Okay.
Yeah, you can share it now.
So let me start. I'm looking where is my PPT in the screen. Okay, so okay. Share desktop one, share. Okay, you see share screen. Now you will see presentation. PowerPoint. Where is the PowerPoint missing now? Just a minute. You are able to see me, my screen, right? So let me.
Yes.
PowerPoint is normally just a minute. Yeah. Yeah, here. Yeah, you are able to see, right?
Yes.
Okay, good.
Yes. Yeah, we can see it now.
Okay, so I will discuss about translational bioinformatics and what we are doing in this. And so primarily we are working on translational on a cancer research. In cancer, what is happening is that in a family, if there is a cancer, you need to identify if the person has certain markers. Those markers has to be identified. So today what is happening, previously what used to happen is that people used to have a set of genes where just they will identify by linkage analysis, and then slowly you can identify within families by sequencing and also the common mutations by GWASs. But then the genome sequencing cost has decreased over a period of time in very rapidly, as you see on the in the blue bar. It costs less than $1,000 today to sequence. So what one can do is that one can take a family members and sequence their genome. So basically I can have my parents genome sequence and my genome sequence and maybe my children genome sequence, and then I can analyze using bioinformatics what kind of diseases I can get. And this is the current one I am showing you in the slide is from colorectal cancer, but one can apply this to all other cancers. So there we are working. So I have tried to generalize it so that audience will understand. And for that we develop methods, and 3 different methods we have developed. And the third method we will call it in the same way or not. In the third method, whatever result we get, we merge them together to see in a pathway context. So one gene product will act on other product and form a kind of a street or pathway. And that method. So I'll go briefly into it. So what we have done so far is that we have developed the pipeline. I'm not going into detail of the pipeline, but we have studied 53 families of different cancers, and 53 families means multiple members. So on the average, each family will have 5 members, husband, wife, and kids. So the total number of human was analyzed in this study was 257, and we identified 15 markers, 51 marker from the 53 families. So there are families where markers are not identifiable. So when we do that, we have, say, for an example, in this family, these two have uh, uh, PTC is a thyroid type of thyroid cancer, and third member has another kind of PTC, so which thyroid cancer. So that is not, that patient will not mix with this patient. So you analyze it, and we pinpoint what is the cause of cancer in this family. Yeah, so one single, you start with a lot of data and you end up with a single one. And here we are showing in the right side, when you look into the gene marker gene, I see that all the green bars means most favorable one, and the most favorable one is then identified to be conserved across different species, not just human. And then if there is a change, that is lethal. And that's what we explain in this paper in 2018. Um, and I just try to be as generic as possible. There are mutations in the cancer, which is, if someone has mutation in that, those are screened, and MMR mutations are of that type. In 3 families of colorectal cancer, we identify new mutations. Uh, so these mutations can be also, and these are European data. So these are coming from Poland. This data is coming from Poland. And these, the first one is never described as large duplication. So basically, you can imagine this big chunk was duplicated, and it is found in 2 control and cases. The case, 2 cases and 1 control. So 1 control is young. So basically, that person may get cancer over when the person is getting aged. And so that in this family, 2 have cancer and 1 is under risk. And similarly, we identified another family, uh, the mutation in this MMR mutation, which we call, is a new one. So these are not reported in these are not in a clinical diagnostics yet, but with our study, it is possible now to have it. And this paper is just recently published. So we get 3 new mutations. So and then we also analyze different families, and I'm trying to give you as generic as possible that these families have high risk of Hodgkin's lymphoma. And then we analyze this data in a different manner, and then similar way we identify genes, and then mutation is located here in the protein, and then we try to characterize in the pathway is network of proteins that mixed together. And these, in this pathway, you see that cancer pathway, developmental pathway, and RNA pathway is activated. And then we try to explain mechanism of DICER1 mutation. So if somebody is biologist, they will understand that if something is happening, then it should not happen in the control, but it should only happen in the cases. So what we see here, to our control, they are going up while cases are going down. So they are difference. And then the last slide, we are trying to correlate with that is our process is not random. And if you take a random one, they behave same way. Okay, so with this, we developed a pipeline for rare variant detection for high-risk cancer family. It can be European family, it can be Indian family, it can be Turkish family, it can be American family. So this is a standard method. One is going to nowadays to the doctor, and doctor says, okay, probably the person has potentially these symptoms. They will note down, and once they note down, they will tell the family to have genome sequencing, and then this is called clinical diagnostic genome sequencing, and then one will take it. So we have developed those methods, and now I have to briefly discuss this with you. And that is the future. So that is not only cancer, one can use in diabetics, one can use in cardiovascular diseases, one can use it for rare diseases which are of different types and different ways one is dealing. And okay, so the overall what we do is that we develop cancer genomics platforms, and we also have roles in we also look for non-coding common pathways related to whether cancer modeling. And then what we do primarily do is the cancer genomics, pipeline development, machine learning based approaches, and open collaborative projects. So that's a whole overall view of our research. So we have also developed different kind of strategies, pipelines, and optimize those pipeline and studies. And this is an example of it. And here we are showing you in a breast cancer result. So breast cancer is of two types: triple negative breast cancer, where the markers are not there. The one is triple negative and one is normal breast cancer. So we want to distinguish the two different type of breast cancer. And there we have analyzed the data, and that's one of the important study we have carried forward in recent time. And then we are applying with the machine learning with Yusuf Malik from Israel, machine learning based method for this analysis. And we have a methodology where we can distinguish between different cancers. So you see the different cancer type here, and in different cancer type you have different mutations, different genes are upregulated. So this is a combinatorial of pathways, and this is coming from machine learning based approach. And then when a student is coming to us, we also give them a smaller projects where we just look for the mutation in a specific gene or protein, and then we collect the data from public resources, try to analyze it, try to figure it out, and then do it. And this is how it looks like when you do it for SMAT4 gene. We have done mutation from different resources and mutational analysis, and you see that MH2 domain has more mutations compared to MH1 domain. But then which one are critical? These most of them are critical one are listed, and they're coming from cancer. They are not coming from the public resources, and quite obvious. These are from the cancer samples. So this study is still going on, and we are trying to evaluate the clinical aspect of these mutations and how one can use, how a person, when the patient is coming has a SMAT4 mutation, can we say this person will get a cancer or not? And that is computational. By computational method, these screenings are getting more and more popular and more used in clinical diagnostics. So we basically what we do is that we develop translational bioinformatics based approaches. We develop bioinformatics data science for focusing on translational cancer research, and we provide A to J translational research approaches. And yeah, I would like to thank you, because I was not aware of the audience. That then I realized that audience will be a generic audience. So what we normally do is to translate bioinformatics into clinical diagnostics. That's our approach actually, and that's what we were working. So I'm open for the question.
Thank you very much.
Yeah.
Dr. Kumar, thank you so much for your contribution. We greatly appreciate it. So if there are any questions or something else you want to add, I guess everything is pretty clear.
Yeah, okay. So you can
Yes.
raise the question if you have later on. Yeah, so have a nice time, and yes, thank you very much.
Thank you. Thank you.
Thank you so
have a great
much,
evening.
Dr. Abhishek Kumar. It was really great to see you. So we hope we can meet in our next event.
Yeah,
Thank you so much. Have a lovely evening. Thank you.
Yeah, thank you. Have a nice
Thank you very much.
Thank you very much.
All right, we have come to the end of today. Right, let me just introduce, let me just make an explanation in English. So we are going to continue tomorrow with the third day of the conference. We will have again, many distinguished speakers from different parts of the world. So I hope to see you tomorrow. Thank you so much. Have a lovely evening.
Evet, yarın görüşmek üzere diyorum ben de. Herhalde biz bize kaldık da.
Evet hocam.
Evet.
Yarın görüşmek üzere.
İyi akşamlar herkese.
İyi akşamlar. Hoşça kalın. Görüşmek üzere. Hoşça kalın.